Announcements. One is that I just sent out an email around the quizzes. So, as many of you have pointed out, answers for quizzes 2 and 3, your answers are not available on D2L. That's because of glitches I encountered with the program dating back to the original snow day. I think I fixed it for quiz 4. You should have your grades for quiz 4. But for quizzes 2 and 3, all the quizzes and answers are online. It's under the exams this class part of the website. And if you have trouble deducing what your score was, if you didn't write down what your answers were, you'd forgotten, just email me and I will email you back your scores for quizzes 2 and 3. Okay. And there'll be a quiz 5 online probably by tomorrow or Wednesday due for next Monday. Also for quiz 4, the first question actually had 3 correct answers to it. So I made not one but 2 mistakes in writing the possible answers for that question. So that's also in the email I just sent you. So I will regrade that question by hand, and if you got any of the 3 possible correct answers, you will get credit for that. The other announcement is that we have the final exam on March 17th. I think that's right. Yes, at 12:30, and we got the full 3 hours for that. So this classroom time was extended. So that's good because it means that nobody will be short of time for the final. Okay, questions? Yeah.
Is the exam written to be like 3 hours, or is it?
It's 160 points, okay. So the midterms are a little less than half that. Okay. So it's like 2 midterms.
Okay.
Yeah. Okay, so today we're going to finish up talking about regulation, and then we will talk a little bit about nitrogen metabolism. Okay. So we went through glycogen regulation with respect to glycogen phosphorylase and glycogen synthase. And just quickly, it'd be easier to just kind of show the slides for these. Phosphorylase is, of course, the enzyme that mobilizes glucose from glycogen. So it is activated when it's phosphorylated, and the phosphorylation of it is done by a kinase that is in turn hormonally activated by glucagon, which is a peptide hormone secreted from the pancreas that signals low blood sugar. That's done when blood sugar is low. So we want to activate phosphorylase and therefore break down glucose when blood sugar is low. And that's in the liver, so the liver is going to take that glucose and send it out to the tissues. In muscle, we have a different hormone, it's epinephrine. Epinephrine can operate on either liver or muscle. It's the fight or flight hormone, so it needs to immediately generate activity in the muscle, so you need energy, so breaking down glycogen that's in muscle is appropriate for that. Right? So either way, we're activating phosphorylase when it's got a phosphate group on it through its kinase, and then it's deactivated by a phosphatase. Right? So there's the hormonal cascade through which glycogen phosphorylase is activated here via this cascade to make more blood glucose. Another mechanism for activating phosphorylase is by binding glucose to it. And so glucose binding it triggers a conformational change, which renders phosphorylase less active, which of course makes sense if there's already a lot of glucose around. You don't want to be breaking down more glycogen. Now, glycogen synthase is the enzyme in the synthesis pathway that is regulated. And in this case, it is inactive when it's phosphorylated and active when it's not phosphorylated. So exactly the opposite way around from glycogen phosphorylase. Two kinases act sequentially to phosphorylate glycogen synthase in order to turn it off. One is the CK2, and the second one is GSK3, which functions processively to phosphorylate 3 consecutive serines spaced 4 or 5 amino acids apart. Right? An insulin cascade operates to phosphorylate GSK3. And when GSK3 is phosphorylated, it recognizes itself. It's kind of fooled into thinking that its own molecule, its own peptide is a substrate. So when the insulin cascade results in a kinase that phosphorylates this, it's actually PKB. GSK3 becomes inactive. When it's active, it operates processively on the substrate glycogen synthase. So that's the inactivation, and the activation is by a phosphatase, which is itself activated by insulin here. So insulin blocks here, activates here. We have high blood sugar. We want to synthesize glycogen because that gets rid of the sugar, puts it up into a storage particle. Right? Lots of glucose will also activate glycogen synthase, glucose 6-phosphate related to glucose. And here the low blood sugar hormones inactivate the phosphatase, right? Because we do not want to be synthesizing glycogen from our scarce glucose if we already don't have very much. Yeah?
Does this happen in both the liver and muscle, or just the liver?
This happens in liver and muscle. So the pathway that's missing in muscle is gluconeogenesis up to glucose. Muscle has both pathways to go from glucose to glycogen and glycogen to glucose. There's differences in muscle and liver that I'll mention in a moment. Okay, so that's how glycogen synthase is regulated. And via insulin, here's insulin cascade operating through PKB, phosphorylate GSK3, and therefore turn off glycogen synthase. Inactivate it, leave it on, so that we make glycogen from excess blood sugar. In that insulin cascade, a side reaction is that the PKB kinase, which is phosphorylating GSK3, also has a side reaction to help the receptor in the membrane take in more glucose. So that's kind of just reinforcing. Which takes us to this slide, where we are associated with a glycogen granule here. And we have insulin coming in, right? And insulin, a protein that's triggered by insulin is called insulin-sensitive kinase. This is a different one than one we've talked about so far. Phosphorylates a protein called GM, and when GM is phosphorylated, it sticks in this complex with glycogen phosphorylase, phosphorylase kinase, and glycogen synthase, where PP1 is also associated. So PP1 is the phosphatase. Right? So insulin has a consequence of by phosphorylating GM, it keeps PP1 in the particle, and therefore PP1 can act on all three of these. So if PP1 dephosphorylates glycogen phosphorylase, then it inactivates it, which is what we want if there's already high blood sugar. It activates glycogen synthase, right, which is also what we want for high blood sugar, and it inactivates phosphorylase kinase, which is also what we want because that will also result in higher blood sugar, which we already have. Right? So this whole thing, when it hangs together, is something that insulin likes. Epinephrine, on the other hand, wants to trigger more release of glucose because we need the glucose for fight or flight response. So in that case, it operates through PKA, and PKA can either function by phosphorylating PP1 and pulling it, by phosphorylating GM and pulling it out of the particle, or by promoting the association of GM with an inhibitor protein, which also pulls PP1 out of the particle. And so when PP1 is out of the particle, none of these reactions happen, and that's good for epinephrine because it means that blood sugar will go up. Right, so these are alternatives, this versus that. Right? Presumably, both of them will not be present at the same time, or else there's some deeper systemic problem with the body metabolism. So, this is really a review, then, of what we've just reviewed and talked about last time. So, high blood sugar, up insulin, more glucose into the cell from a membrane protein that lets glucose in. That insulin-sensitive protein kinase on the last slide activating PP1 and having these effects that we just discussed on those 3 enzymes. Okay? Activating PKB to take out GSK3 to activate glycogen synthase. Higher insulin through those cascades in the primary glycolysis enzymes, leading to greater glycolysis. So high blood sugar, greater glycolysis, greater glycogen synthesis, and more glycogen, and less glycogen breakdown. And it's the opposite on this end here. Not all of these cascades were shown in specific slides. So low blood sugar, glucagon, higher cyclic AMP, higher pKa, we saw that. Higher pKa results in lower levels of glycogen synthase through mechanisms we didn't see on one of the other slides. But the upshot of it is that glycogen synthesis is going to go down when blood glucose is low. Right? Glycolysis will go down, and glycogen breakdown will go up. So another summary slide is here. And now we're looking at, a little bit more specifically, also the differences between liver and muscle. So glucagon is operating on liver. It does not operate on muscle. Okay. When it's signaling low blood sugar in liver, and it gets to liver, it leads to the breakdown of glycogen, and therefore the export of blood glucose. Right? So liver's here on the left. So we know liver wants to let its glucose out. It stimulates gluconeogenesis up from pyruvate, also leading to glucose out.